talking about drug development process. I should begin by talking about what Mark Twain had to say about our country. On his sojourn from following the equator, he says, "So far as I am able to judge, nothing has been left undone either by man or nature to make India the most extraordinary country that the sun visits on his rounds. Nothing seems to have been forgotten, nothing overlooked. And no longer are we a country of snake charmers, camel riders, since we in India are making rapid progress." And if one has to progress, obviously one has to have a vision. And this, I suppose, is the vision, a glimpse of India's pharma future, as we say, where if you are currently out here in terms of 2004, 2005, 2007, this is where we propose to go. We would be lead players in new chemical entity research. We would have contract research and work on high-end contract research. We will be in clinical research as a preferred partner, approximately doing 10% of global clinical research work. And if that be the case, can we have an hypothesis where India will leapfrog to the next level of drug discovery, moving as we did from a variety? Can we get across to around 2010 future of medicine? And if India should start aiming towards this, I would say, thanks to our hop, skip and jump on the Indian Odyssey in terms of patents, we took that hop in 1999, the skip in 2002, and jump in 2004. And as of today, pharmaceutical products are patentable, which would mean India has to start looking at its own drug research, its own R&D. And if we have to do that, and if we look at what the government has been doing, well, we had product patents, step 1, financial incentives for innovators being given, price increases for controlled drugs, ending price controls, and if between 2005 2010, if we have made all of these progresses, I think we are essentially looking at how we can go ahead and look at the economic fundamentals of new drug development. And if you look at how in the earlier period, the current model spoke in terms of 10 to 12 years for drug development, it can perhaps be shortened to anything between 3 and 5 years. We went ahead and spent during the pre-launch approximately 200 million. You would have success rate from entry into man, which is about 50% or better with new drug development. And if one talks in terms of new drug development, one will talk in terms of what have been the approaches to drug discovery in the past. Historically, if you look at, you had variety of plant products like cinchona, quinine, which came from a cinchona tree, the willow bark, which gave us aspirin. We have studied disease processes and looked at how, say, for a breast cancer, you came out with tamoxifen, how you came out with Parkinson's disease, understanding the mechanism there and coming out with the L-DOPA. Studying the biochemical and physiological pathways, how we could come across and identify renin-angiotensin as a mechanism and therefore anti-renin or anti-angiotensin drugs. Look at how pharmacology has helped us and look at structure-activity relationships and of natural compounds like a variety, or look at the beta adrenoreceptor drugs like propranolol and how we selectively moved from propranolol to atenolol. Design to fit known structurally identified biological site, and that's how you came out with angiotensin converting enzyme inhibitors. And of course, when you talk in terms of serendipity, random screening, we came out with a variety of drugs like penicillin, dimenhydramine and pethidine. Today, of course, we will talk in terms of genomics and how identification of receptors, gene therapy and recombinant material is possible. And now if one talks in terms of drug discovery, I would say necessity is the mother of invention, and therefore the driver there is unmet medical need in a viable market. Taking coal to Newcastle, as they say, but then the source of drugs are animal, plant, inorganic and synthetic, and I'm sure we remember some of those sources of drugs. But we will go back to the time we were created and talk in terms of how man is a valetudinary animal. always concerned about the state of his own health. Concerned as he was about the state of his own health, if you look at the history of drugs, it perhaps is the history of mankind. And if one talks in terms of how primitive man experimented with plants that grew around him, it's that experimentation that has given us most of our drugs of today. And you can go back and talk in terms of how reserpine first came, how digoxin first came, atropine, aspirin, and you name it. And even today, to standing here out here, I can say you still look at paclitaxel coming from a plant source, which is Taxol brevifolia. So we are still looking at a variety of natural sources for obtaining some of our modern day medicines. And if you talk in terms of how man is trying to ward off death, you're looking at modern science and technology with its marvels making us live, if not forever, at least much longer. And if one is looking at how drug discovery and development have to be looked at and ask that question, why know about drug discovery? The answer there would be, we need to be aware of how, why new drugs are discovered, know the processes involved in drug discovery and development, see where pharmacologists like Dr. Chanda Kulkarni and Nitya and other biosynthesis contribute, know about the difficulties and dangers inherent in the drug development process. If you go back to the past, and in the 1800s, we had natural sources with limited possibilities. You had individuals preparing these small scale drugs, which were not purified, not standardized or tested, had only limited administration capabilities. There were no controls, no idea of the mechanism of action, and start looking at how in the 1900s, in the 1990s, you now have synthetic source of drugs, unlimited possibilities, prepared by a variety of pharmaceutical companies. On a massive scale, you have highly purified drugs, standardized and tested, worldwide administration with tight legislative control, and of course, mechanisms of their action partly understood. If one talks in terms of why are new drugs needed, and one will talk in terms of unmet medical needs, new diseases, downstream health costs, cost of therapy, cost to individual country, and sustain industrial activity as some of the reasons why new drugs are needed. If one exactly gets to the core of activity, one would start talking in terms of, look, how long it would take. It would take perhaps 12 to 16 years before a new chemical entity can get into the market. And as we begin with 5,000 to 10,000 products that get screened, put them through a funnel, you end up with 250 products that could enter preclinical testing, 5 that enter clinical testing, and 1 gets approved by the US FDA, for that matter, the drug control authorities. And if such a long process, drug development is risky and indeed expensive, costs you approximately about $1 billion or $1 billion plus in terms of getting across a drug into the market. And if one is talking in terms of the phases, you look at starting there, it takes approximately so long for us, about 15 years, $1.2 billion US, like I mentioned to you, and you will have phases of lead, drug discovery, look at preclinical development, clinical development, before you get across the drug to be in the marketplace. If one is looking at how do we start looking at drugs, one will talk in terms of how you target diseases and start looking at diseases that afflict man as of now, most commonly central nervous system diseases like Alzheimer's, Parkinson's, which have defeated man as of now being areas of focus for drug discovery. And as you come along the line, infectious disease, I mean, you're just talking about AIDS, you have oncology, a lot of malignancies that still bug us, which have defeated therapy. Metabolic diseases, and if you look at India, we have double trouble. We not only have diabetes, you also have a lot of heart disease. Every 8th person in India has diabetes as of now, and therefore you're looking at metabolic disease, ophthalmology, urology, women's health and inflammation as diseases that are targeted for newer drug discovery. And if one talks in terms of the process, one starts with a lead compound selection, which could be a process that begins with random screening of a variety of products, which gets across to the next step, which is combinatorial chemistry with high throughput screening, targeted synthesis, drug modeling, and of course, human genome mapping as of now. If one is talking in terms of combinatorial chemistry with the IRORI system, we anticipate 10,000 compounds will take about 2 months with 1 or 2 people. That's what's combinatorial chemistry. You have your rational drug design, high throughput screening, all of these processes trying to help to cut down the time required to get across to a lead compound. And if one is talking in terms of the expense, I did talk to you about the fact that it's 1.2 billion US, time consuming, cumbersome, and if 1 in 10,000, 1 lakh products gets 10,000 products gets across to be a new chemical entity, ultimately my marketing friends will say, is it marketable and what will the outcome be? When you move across from lead development to discovery testing, what we are interested is, does the drug work, and therefore look at animal testing. Is it long or short acting? How does it work, and look at its specificity of action, and finally look at oral administration being possible. Costs you a few million US dollars to be doing this job. And if one is talking in terms of how lead discovery as a part of preclinical research, new product development takes up as clinical development, we need to prove 3 things. Is the new drug safe? Is it effective for a specific disease? And can it be manufactured with CGMP facilities? And is this process reproducible? And among the discovery research areas, you have a variety of functions that people can play. Having found that a compound is initially useful, what the synthetic chemist would try and do is find out if chemical synthesis and scale up is possible. And when one is looking at chemical synthesis and scale up, one is looking at large scale manufacturing of the bulk and formulations, safety of the environment and people involved in manufacturing, looking at the patent issues, and once you have a hang of the patent issues, look at financial considerations and see if it's going to be meaningful to get this new product into the market. And one would go on then to start looking at chemical and biological characterization. By chemical characterization, you would look at the structure, the synthesis, purity, blah, blah, blah of the drug. When you start looking at biological, not only would you look at the pharmacological profile, the LD-ED50s, you would start looking at different activities, that biological activities that this drug has. And if one is talking of biology, one is looking at biology replacing chemistry as of now. If synthetic chemists had their say 100 years ago till today, perhaps the future is in life sciences. And in the new millennium, biology has replaced chemistry as a leading science with biotechnology joining hands. And of course, with information technology joining hands again, what really is happening is they are guiding us through the library of life. So one is looking at how biotechnology products have slowly overtaken synthetic chemistry products in drug research. Irrespective of whether it's synthetic chemists or biologists who have helped us get the new chemical entity across, what one needs to do is the new chemical entity stable on exposure to light, moisture and temperature? The next step, can it be formulated into small tablets and capsules? And finally, if you wish to administer it to young children, is it dispersible? Costs you some money to be doing these studies. Now, when we move on and start looking at the pharmacokinetics and pharmacodynamics, what the drug does to the body, what the body does to the drug, start looking at the side effects and toxicities, and during animal studies, correct the early absorption and toxicity problems before submitting an investigational new drug application to the FDA. What one has to do is get across and conduct animal studies. And if you talk of animal studies, I found this a very interesting photograph there. I happen to be a Bush basher, and I found George Bush somehow has not said anything about animal testing as yet. He has spoken enough about stem cells, cloning and whatever. And since I found this interesting, I said, there is George Uncle Bush patting on a rat and saying, I will continue to permit animal toxicology studies to go on, and therefore you do acute toxicology, subacute toxicology, chronic toxicology studies lifetime toxicology, reproductive studies, normally in 2 rodent species, later on in bigger animals, as animal testing of drugs prior to its first use in man. We also of course compile data about the teratogenic, the genotoxic and the carcinogenic potential of drugs. Among the animal models of efficacy, one would start looking at existing normal behaviors, effects, anesthesia, contraception, paralysis that the animals undergo. Try and create behavior. So, you have obese rats, you have hypertensive rats, you have anxious rats, you have epileptic rats, you have diabetic rats. So, you can create models on which you can start looking at how drugs behave and find unrelated behavior affected by existing drugs. Now, you can design studies which are an exact replica and therefore have 100% predictive value for such tests. When you have drugs that have similar mechanism of action, you would say they are a fairly good predictor of what will happen in mankind. And of course, when the mechanism of how a drug works on the rat and how it works differently, say on the human, differs. I mean, if you look at the fact that I and you have been swallowing a lot of metronidazole, metronidazole has turned out to be carcinogenic in rat intestine. I and you have been exposed to tons and tons of metronidazole, hoping for sure that we don't have rat intestine in our own intestine, and therefore we won't have carcinogenicity. Like I mentioned to you earlier, acute toxicity, chronic toxicity, I won't spend too much time since this is animal toxicity. We try and design a situation where we have known all of the preclinical information, and that's when you submit a document to the FDA saying, I wish to conduct clinical studies with the new chemical entity or the investigational new drug, and that's when you design studies that normally go in 3 phases, 1, 2 and 3. Depending on the success with which you finish your phase 1, you get across and do a phase 2. Should you be unsuccessful, you go back 2 steps, or you go back to the animal toxicology all over again. So, if one is talking in terms of how research is regulated in humans, I don't have to spend time on this. I am sure you are aware of the fact that all of whatever we do in humans has to be done as per the ICH GCP norms and should follow good clinical practice guidelines. So, if one has written a protocol for a phase 1 study, our aim here is to check if the drug really works. If it works, is it safe, and how does it behave in humans? The aim of course is to ascertain safety, identify the range of doses tolerated, to look at the absorption, distribution, metabolism and excretion of the drug, look at a study population which are normal healthy volunteers. So, you would start with, since it is looking at safety, look at 30 to 50 normal healthy volunteers in whom the drug is chosen as an extrapolation of the animal dose to get across to be used at doses which are either escalated or de-escalated, and therefore identify a right dose that can be used. Should a drug pass through this stage, it gets on to a phase 2, where our interest is to find out, is the drug really efficacious in patients, and therefore this is perhaps the first time patients are exposed to get this drug, and you will have a first look at the efficacy while you continue to look at the safety. You would design a placebo controlled, randomized or a double blind study. I am sure you will be exposed to some of this as you go across in the program, and at select medical research centers where they have facilities to conduct a foolproof phase 2 study. Approximately 200 to 300 patients suffering from a target illness, depending on the therapeutic segment that you are trying, are examined. Takes as long as 2 years to complete a foolproof phase 2 study, and a decision on whether to go ahead or not go ahead is made based on a phase 2 study. Phase 3 perhaps is an extension of a phase 2 study, but in larger patient numbers to establish safety and efficacy in large patient groups. Again, can be designed as a randomized, controlled, either placebo or controlled drug, double blind study, conducted at several hospitals, multi-centric, multinational studies or whatever, and may take as long as 3 years for us to complete collection and collation of all data from a phase 3 study. And once we have done this, we can put together, I am sure you will get exposed to this, a controlled and uncontrolled study, open or blind, parallel, you can have sequential, you can have a crossover study, you can have a variety of designs. So once we have completed all of these, here you have 120,000 pages of data. As of now, you can electronically submit this document to the US FDA. May take them approximately 12 months to review, and when they get back to you, the answer is not a definite yes or a definite no, but they will probably come out with indications that are permitted. They talk in terms of limitations, ask for a phase 4 study, and then start looking at chemical testing of this drug to ensure that the chemical compound that you have set that works really works. This then is a colorful depiction of what happens in the US FDA. There is that preclinical research, how you pass through the phases of phase 1 to 3, how couple of occasions the US FDA would permit accelerated development for certain drugs, for certain orphan drugs that we will talk about subsequently, and how couple of occasions drugs can be rushed into the market if it's of great importance. So that's one there. The next step there, where one is talking in terms of the steps that you would take when you make a new drug application. Once you have data from your preclinical study, how from a new chemical entity you move to a investigational new drug and did the preclinical studies, how from the preclinical studies you would get on to a phase 1 study based on a new drug application, and how you would get that across to approval. Now, when phase 4 studies are asked for, essentially phase 4 studies are looked at as post-marketing surveillance studies in the general population, and ideally it should have the similar rigor as all phases of the study. Should start looking at an ICH GCP based design for phase 4s. Unfortunately, in this country we are giving a go by on this. We must start looking at how approved drugs can end up with newer indications. I mean, incidentally, when we had metformin that's essentially used for diabetes, we found that metformin did well in women who had polycystic ovarian disease, and therefore metformin became to be useful in polycystic ovarian disease. How a drug that was given up in the past, thalidomide, has come back into clinical medicine, again based on phase 4 observation. And of course, normally withdrawals are based on pharmacovigilance and undue toxicity. And of course, we have the recent incidents of Vioxx that was withdrawn from the market thanks to a pharmacovigilance study. And of course, rosiglitazone, which is producing a lot of edema and the swelling in patients who have diabetes. So phase 4 would obviously try and help us try and get certain drugs out of the market if they are producing undue toxicity, get to use certain drugs for extra indications that may be useful. Now, when one talks in terms of post-registration monitoring of drugs and how crucial that is for drug development, one is talking in terms of the yellow card system that prevails in the Western world, intensive monitoring of defined groups, restricted release, and of course to monitor incidence of disease problems. India slowly is a signatory, in fact slowly but surely is a signatory to pharmacovigilance as of now, and over a period of time I am sure we also would have the similar rigor as the Western world has with regards to ensuring that we have safe drugs. Regulatory process differs from country to country, demands our safety and quality of product, encourages efficacy and need for a product, grants clinical trials certificate if volunteer and animal data are okay, approves protocols, contains a variety of, like I said, volumes and volumes of data. Original data is available, and of course, it's a two-way process where the regulatory authorities are trying to look at what sort of process the drug has gone through, and as a sponsor, the companies are looking at how we can ensure we have safe drugs enter the market. If I would summarize new drug development and say starting off from the laboratory through a process of lead compound selection, discovery testing, chemical synthesis and scale up, say stability and formulation development, animal studies, human trials, and to get a drug into the market, would take us as long as anything between 1 to 12 years. Start looking at 10,000 products getting into the funnel on the top, and finally one product coming out as a new chemical entity that's workable. Look at approximately 800 million US dollars for certain, 1.2 billion dollars for certain others as revenue that is getting spent. And we could ask one question, which is, if it's an expensive, time consuming process with numerous bottlenecks, can we have alternates to drug discovery? Can we start looking at, if this process of target identification to clinical trials and getting to drug to the market is so costly, do we have an economical, time sparing model with least bottlenecks? And it's here I must compliment the efforts put in by Dr. Mashelka, the former director of CSIR. And I remember talking about reverse pharmacology at that point in time, about 4 or 5 years ago, when we said, can we go back and look at clinical trials, preclinical studies, relevant science, and then start looking at large scale trials? I mean, is it okay for us to be talking of reverse pharmacology? And as we were talking about this, I remember going back to him and saying, so why don't we look at the rich cultural heritage we Indians seem to have? When Lord Brahma created us and spoke to Dakshahati in Ayurveda, he spoke with him and said, Anadim avyayam divyam vyapakam vishwabhishijam trikala baditam nityam ayurveda upasmahe. When he said that, he said, nityam ayurveda upasmahe would mean, if you have trouble all 3 times of your day, which is morning, evening and night, when you suffer from sickness, nityam, every day, practice Ayurveda. And when God is supposed to have said that, he said, look, all medication came into this world from primitive man's experiments with plants that grew around him. When I initially decided that I would create you, I probably also brought in the concept of anugraha, which is blessing. If I created a snake and forbid you from meddling with that snake, I also created, and you went ahead and meddled with that snake, got bitten, I created a plant that perhaps would give you benefit from, I would say, that would prevent you from dying of snake bite. So anugraha is God's grace or blessing, and out today we can talk in terms of anugraha being serendipity as modern men. If that apple had not decided to fall down, perhaps Isaac Newton would not have had the law of gravity. So one is God's grace or serendipity. The second one is anumana. When we had initial medicine, people had suspicion, people had a hypothesis, and that's today's hypothesis, where we said that particular plant perhaps is useful for XYZ. And that's how people came out with the concept of a plant which is Adatoda vasica. Now, for you and me, Adatoda vasica looks like a botanical name. But if one is a Malayali and understands Ada is goat, Toda does not touch, Vasica is a plant. And if it's a plant that a goat refuses to take, it's perhaps, and is a creation of God, it's perhaps basically because that was bitter. When man went and tested that, he had a suspicion it might work. When he tested it, he found out that is the best mucolytic. You have bromhexine today. I think most of our medications, the plant-based medications, the animal-based medications, have come from Anumana. Now, Anubhava is experience, and I'm sure our ancient rishis and wise men had a lot of experience. They had what's called Anubhava Gnanam, a lot of experienced knowledge, and it's based on their experience, the experience that they wrote together in a variety of treatises. I mean, you go back to Charaka Samhita, you will find a treatise on how exactly a variety of new medicines could be made. And that's perhaps what is Anubhava of yesteryears, which is equivalent to experience of today. And finally, if one talks in terms of how Anusandhana, which is putting together those various experiences, which today perhaps would be an equivalent for meta-analysis that we do for a variety of studies. So if you look at the past and the future, the present being the dividing line between the two, there is a definite semblance of order, semblance of similarity between what happened in the past and what's happening now when you're looking at trying to get a few new drugs. Is this only culture? Is this only Sanskrit? No. The Indian experience has seen us get 2 antitubercular drugs which are in phase 2 trials, and if I'm not mistaken, Lupin is conducting research. And therefore, I think we in India have now woken up to the fact that having signed the Patents Act, having a problem of no longer being able to be copycats, we are now forced to look at necessity as a mother of invention for our foray into drug development. The future of drug development, perhaps one would start looking at, are there third world diseases for which the Western world is not interested in making medications? I mean, the Western world is not very keen on looking at what's happening in malaria, basically because they don't have much of malaria. Are there orphan drugs with few users? How do we improve safety and efficacy records? How, if by chance Maneka Gandhi happens to be the Prime Minister of this country in the next election, how will we reduce animal utilization? I mean, do we have an opportunity where instead of using animals, can we start using cell lines? Can we have early human volunteers? I mean, I have been foraying into stem cells as of now. How would you like it if we start producing a few hepatocytes, a few cardiomyocytes, a few neuronal cells, and directly test drugs on that and say, can I hasten the process of drug discovery? Perhaps the future will see that. How would you look at new diseases? How would you start looking at new biology that we are looking at? How would you look at clone human receptors, disease model by gene? And how would we start looking at patent times and increasing cost? So this perhaps is what will drive the future of drug development. And if India continues with the same stance that it is, this is perhaps the Indian pharmaceutical industry and its road ahead. November 2010, The Economist will perhaps talk in terms of India, the new global pharmaceutical superpower. We perhaps could have this sort of a cover page, our knowledge with genomics, our Indian patient leading from the front. Hopefully, this is a guy who's inspired by a saying from the Bhagavad Gita. How I and you will start looking at, if we have known the sequence of the genome, how would we then sit down there and start looking at climbing steps, climbing the ladder, start looking at newer drugs? How, in order to make this happen, we should start looking at global outsourcing and partnering opportunities. A friend of mine from Eli Lilly, where I used to work, talks in terms of strategic alliances not only have a future in the pharmaceutical sector, they are the sector's future. And therefore, a lot of the Western world is looking at how they could make use of Indian clinical research, contract research potential to sort of get across to new drugs. And finally, if one is talking in terms of the future of drug discovery and development of innovative therapeutics 2007, one is talking in terms of innovation in stem cells, RNA delivery, protein-protein interaction, experimentation, requirement of complexity, speed and integration. One is talking in terms of commoditization, looking at efficiency and reduction in cost. One is looking at 4 core areas: oncology, central nervous system disease, metabolic and inflammatory disease as diseases that we need to tackle, and are looking at novel targets, animal models, biomarkers, surrogate markers, whatnot. One is very closely looking at how stem cells would come in as an alternate to drug therapy in the near future. And if this is the future, I thought I will probably take that up in the next session that I have, and kindle your interest by saying, we in India were in the Stone Age. I think we have now moved to the Clone Age. And there is every likelihood that if we keep pace with what really has been happening in the drug development area and start leading from the front, and I did talk in terms of how the Lord very clearly said, yad akruti shreshtas tad tad lokas anuvartate, which is, if as leaders of men, we in India decide to lead from the front, the rest of the world will follow. And we in India, I am sure, are capable of doing that. I can answer questions now.
What do you think the impact of this shift of patent from process to product on the on our country, especially in the context of plenty of people below poverty line and those who, in terms of affordability? I would
all right.
Welcome your comments.
In terms of kindling the interest of the pharmaceutical industry, and in terms of exciting us to work with our own drug development, signing patents has been a great thing for us in India. If we had not signed that, we had not made product patentable, what we would have continued is to ape the West, still look at reverse pharmacology, or if not reverse pharmacology, still look at reverse engineering, get across a product. I remember when I brought in omeprazole into this country, I remember the trouble we had gone through in AstraZeneca to get the product. But even before I had filed my document to the DCGI, DRL came out with a product. If I had 10 steps, they had 12 steps, and therefore got away with introducing omeprazole into the Indian market much faster than the Western world, or much faster than the innovator company could do. So in terms of kindling the interest, kindling the enthusiasm, and if I could use a word, kicking the backside of us Indians to start looking at drug development, signing the patent is indeed a wonderful thing. And if that wonderful thing has kindled our interest and we started looking at those two products from our own natural resources, I think it's excellent. If I take the opposite view and say, since we have signed the patent, we would find most of our drugs getting costlier, and therefore affordability would be an issue. Well, look at the phenomenal population we have, 1.2 billion, is it? 1.2 billion. And if you have reached that sort of population, tomorrow if you find a drug that's useful into this country, and we go through the whole process of its registration. I mean, if I were to have done that in the past in my multinational, in a time when I was there, I would say, looking at the scale of operations, we still, and for that matter, India and China have enjoyed preferential pricing, which would permit multinational companies to launch the same drug into this country at a much lower price. Because of the numbers we have, obviously, we could say we will give you more volumes and therefore give us the drugs at a cheaper rate. So that perhaps would happen. And it's a question of time when, if you look at some of those newer drugs and you really make a critical analysis of some of the newer drugs which are looking at unmet medical needs, I think we already have some which we can do well with. I mean, your penicillin still works, so you don't require a 4th generation penicillin to tackle an infection that's as of today relevant. So, I mean, if you look at an MRSA, you still can go back to a penicillin and still look at its, I would put it as logical use. So, you really don't have to depend on the West and say, since drugs are going to become costly, would we in India be able to afford it? The opposite would be, since we have huge numbers, perhaps the benevolence of Belinda Gates or whatever Gates would get us those drugs for use in this country. I don't know if I have answered your question in the way you want it, but I suppose that I think is one of the ways we would look at it.
If we consider the basis of reverse pharmacology, do you think that for development of a new drug, it will reduce the time span and the money spent in course of the process on which already the drug development process is there? Is it one of the bases of reverse pharmacology?
No, it's not a cut short time. It's not a cut short time that we are looking at the age-old benefits of a variety of our traditional medicines. Reason why we are looking at traditional medicines is we have been using, I mean, if I and you had a sore throat and our grandmother gave you tulsi leaves, ground or whatever, and that did relieve your sore throat, is there a background, is there a chemical entity in the tulsi that is useful, safe in sore throat? So what we are looking at is, if these plant products had their benefit, can we go ahead and scientifically look at if they really do have, can we register them as an active compound? I can give an example of, I'm sure if Urmila is back here for a lecture, you can ask Dr. Urmila that day, where we've been looking at, is Brahmi useful? If Brahmi is useful, can you scientifically look at it based on a thoroughly organized phase 1, phase 2, phase 3 work? If Brahmi is indeed useful, can you look at its chemical ingredient and rush through these steps? Now, you don't have to go back to, say, animal studies. You probably will start off with a large phase 3 study, the way Brahmi should ideally be used. I mean, if I were to go back to another example, Charaka Samhita talks in terms of Narikela Lavana. After all, Narikela Lavana is what? A coconut. Okay. Get into the eye of the coconut, drain all the water, put rock salt, Saindra Lavana. Alright, that's what Charaka wrote. And let this coconut with rock salt get cooked over 42 days on coal, red hot coal. At the end of 42 days, if you mashed this or powdered this and administered it to patients, perhaps they had the best of response for patients who had peptic hyperacidity. Not even omeprazole can match that sort of, I would put it as mechanism of action, safety and efficacy. The only problem comes when we said, okay, if Narikela Lavana is used and they put that for 42 days at red hot cold temperature, what we modern men seem to have done is 42 days, 100 degrees, 4200 degrees. So take coconut, irrespective of where it comes from, mash that. You don't get rock salt, you put common salt, and cook it over a day, 20 minutes, 4200 degrees temperature, and mash that powder, administer that as Narikela Lavana. It doesn't work. But all I am saying is, while not taking a shortcut, can we, with our modern efforts of ensuring that you pick the right coconut, if you have to make a Chyavanprash, pick the right gooseberry, ensure that it's standardized and say you'll only pick it up from the south of India, those western Ghats, whatever. Ensure that you have a prefixed standard operating procedure and then say this is exactly the process it will go through for it to be made into Chyavanprash. This is exactly how it will get across into a phase 1 study, ICH GCP norms, whatever, before we go ahead and announce results. I'm sure we could succeed. Thank you for your patient hearing.